Hello and welcome to this Infection Basics video. I'm Dr. Aled Roberts, and in this video we're going to look at how bacteria cause disease using toxins. And so a basic question we should ask ourselves is, what are toxins? At its simplest, toxins are specialized chemicals that have a harmful or even poisonous effect on other organisms, meaning they disrupt normal physiological process, often in very specific and targeted ways. But that definition immediately raises further questions. What does it really mean for something to be poisonous? Well, a poisonous substance is one that can cause illness or death in a living organism, typically when it is ingested, inhaled, or absorbed. Now, many substances fit that description. However, not all poisonous substances are toxins, and this is where the distinction becomes important. To be classified as a toxin, the substance must be produced by a living organism, which in the context of this video means toxins synthesized by bacteria, and so while many chemicals can be poisonous, only those of biological origin fall under the definition of toxins. Now, an important nuance here is that toxicity is not an absolute property. To a certain extent, almost anything can be poisonous. However, it is the dose that determines the overall effect. And it was the famous Paracelsus, often described as the father of toxicology, who famously stated that the dose makes the poison. In other words, any substance can be toxic at high enough concentrations, including things we normally consider essential, such as water and oxygen. And this principle becomes particularly important when we start thinking about how bacterial toxins exert their effects and how even very small amounts can have profound biological consequences on the host. Now, we can't talk about toxins without introducing the concept of toxigenicity, and so in simple terms, toxigenicity refers to the ability of a microorganism to produce toxins that contribute to disease. It describes how a microorganism disrupts normal physiological process or causes damage to the host cells and tissues through toxin production. Now, importantly, toxigenicity is not a fixed or uniform property. Some microorganisms have low toxigenicity. They may produce only small amounts of toxin, or the toxins they do produce may have relatively mild biological effects. And at the other end of the spectrum are microorganisms with high toxigenicity, which either produce large quantities of toxin or produce toxins that are extremely potent, even at very low concentrations. Now, an important point to recognize is that toxigenicity depends on three key factors: the characteristics of the toxin, the host in which the toxin is acting, and the concentration of the toxin within the host. And these three factors interact with one another to determine whether exposure results in mild symptoms, severe disease, or no observable effect. And so let's look at these in more detail, first with the toxin itself. And so different toxins vary in their inherent properties, including their molecular structure, stability, enzymatic activity, and mechanism of action. And these features will determine how a toxin interacts with host targets and how much disruption it can cause at a cellular and systemic level. And as we've talked about, some toxins are highly potent and can exert profound effects at low concentrations, whilst others require much higher quantities to produce measurable effects. However, toxin potency alone is not sufficient. The host also plays a critical role. For a toxin to exert its effect, the host must express the appropriate receptor or molecular targets, and the relevant cells or tissues must be susceptible to damage. And so differences in species, genetics, age, immune status, and physiology all influence how a host might respond to toxin exposure, and as a result, the same toxin can cause severe disease in one host but have little or no effect in another. And finally, even a potent toxin acting in a susceptible host will have little effect unless it reaches an effective concentration at its site of action. And so this depends on factors such as bacterial concentrations, absorption rates, metabolic activities, and the presence of any antitoxins. Now, only when a sufficient concentration accumulates at the target site will measurable biological effects occur. And so together, these three factors, properties of toxins, host susceptibility, and the effective concentration, form the foundation for understanding dose-response relationships. And so when we talk about toxins in a clinical context, we need to be mindful of a few key terms. The first is the TD₅₀, or the median toxic dose. This refers to the dose of a substance required to produce a defined toxic effect in 50% of a population. And importantly, this does not mean death, but a measurable adverse effect, such as tissue damage or impaired physiological function. Then we have the LD₅₀, or the median lethal dose, which is the dose required to cause death in 50% of a test population. And while it has been historically used in toxicology, its use today is more limited due to ethical considerations and the availability of alternative methods. And finally, we have the NOAEL, which stands for the No Observed Adverse Effect Level. This is the highest dose at which no statistically or biologically significant harmful effects are detected compared to control. And this is particularly important for risk assessment, as it helps define exposure levels that are considered safe. And so together, the TD₅₀, LD₅₀, and the NOAEL allow us to compare toxin potency, understand dose-response relationships, and assess risk in a structured and quantitative way. And this has allowed us to use some of the most harmful bacterial toxins, such as the botulinum toxin, as a beautitherapy commonly referred to as Botox. Again, this emphasizes that the dose really does make the poison. Now, when it comes to bacteria, they produce a wide range of toxins and can be characterized in a number of different ways. However, there are generally two broad categories we use based on how they interact with the host. And so the first major group of bacterial toxins are the endotoxins. These are surface-associated molecules, specifically lipopolysaccharides, found in the outer membrane of gram-negative bacteria. Now, because of this, endotoxins are not actively secreted. Instead, they are released when bacterial cells divide or undergo lysis. And once released, they bind to host cell receptors, particularly those on immune cells, triggering inflammatory responses that are responsible for many of their biological effects. Now, the second major group are the exotoxins. Now, unlike endotoxins, exotoxins are proteinaceous compounds that are actively produced by both gram-positive and gram-negative. They are either secreted into the surrounding environment or, in some cases, directly injected into target host cells. Now, our exotoxins are highly specific in how they act and which cells they target, often causing distinctive symptoms associated with a particular bacterial infection. Now, our exotoxins can actually be further subdivided, and one of the most useful ways is based on how they work and cause effect. And so we have membrane disrupting toxins, which create pores in the host cell membrane or destabilize the host cell membrane, leading to a loss of membrane integrity and ultimately cell lysis. Next, we have the AB toxins, which have a characteristic two-component system which consists of an active A subunit and a binding B subunit, where the B subunit covers host cell attachment and entry, allowing the A subunit to disrupt important cellular processes. Then we have sort of effector toxins, which are delivered directly into host cells, often via specialized injection systems, where they then interfere with various intracellular biochemical pathways. And finally, we have the superantigens, which bypass conventional antigen processing by non-specifically activating the immune system, triggering mass cytokine release, which can have fatal consequences. Now, in addition to these sort of mechanistic classifications, they can also be classified by the primary tissue or system that they affect. Neurotoxins target the nervous system, hemotoxins act on blood cells or the vascular tissue, nephrotoxins damage the kidneys, and enterotoxins act on the gastrointestinal tract. And we can bring all of this together, so it's possible to have an AB enterotoxin such as the cholera toxin. And so to conclude, these endotoxins of the gram-negatives and exotoxins produced by many different species and strains are key tools used by bacteria once an infection has been established, enabling them to disrupt the host physiology, damage tissue, and ultimately drive the development of disease within a host. And with that, we come to the end of this video. I hope you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.